As you probably guessed by looking around you, you see nothing. There is nothing to see. The system appears to be frozen, so they're on their way down, and we'll hopefully be here in a minute. That means I've got to talk about a very visual lecture without any very visual things to work with. So this will test my ability as an instructor today to communicate, right? So there are some things I can talk about with respect to protein structure, which is really what we're diving into, that you don't have to see a picture of to understand. And those are the things I'm going to start in talking about, and then hopefully they'll get this system functioning while I'm talking. If they're here, please come to the front, because I think the problem is right here. They said they'd be here in a minute. I don't see them. Okay. Before I start, any questions, comments? Couple things, couple of announcements. One was somebody said, when's your office hours? And I meant to send out an email, and I forgot to do it. Let me tell you briefly. My office hours are anytime I'm not in a meeting, I'm in my office. I will send the class a URL of my calendar so you can see when I'm in a meeting. And you're always welcome to come by. And you don't need to make an appointment. If you make an appointment, I assure you I will be there. But you don't need to make an appointment. A lot of people are just coming by and talking, and I'm happy to do that. So please feel free to come by and ask questions as appropriate. My office is ALS 2145. Excuse me. And I've seen quite a few of you already, which has been very encouraging to me. Don't see much happening. Okay. All right. So that's one thing. And the other is we're turning our attention now from smaller molecules like amino acids that we went through some structural things with last time. And now we're going to start thinking about putting those together into bigger structures. So, of course, when we put together two amino acids, we join them together and they form a covalent bond. And that covalent bond that we make in joining two amino acids is known, of course, as a peptide bond. So the peptide bond is the fundamental bond stabilizing the primary structure of a protein. Now I'm going to talk about, excuse me, I'm going to talk about protein structure at 4 different levels. The first level is the primary structure. And the primary structure of a protein is very simple. It's simple to tell you what it is. It's not simple in terms of what it is. It's the sequence of amino acids that make up a protein. That's what the primary structure of a protein is. The sequence of amino acids that make up a protein. All right? We have 20 different amino acids, and we realize that we have an incredible number of proteins that can be made with 20 amino acids. If I have just 2 amino acids, there's 400 different combinations. There's 3 amino acids, I have 8,000 combinations, right? It starts getting pretty big, right? So we have an enormous possible number of combinations, and every different combination that we have gives a different structure, an overall structure, okay? So I'm thinking now about the overall big structure of a protein. As I change the primary structure, the primary sequence, I use those terms interchangeably. As I change the primary structure of a protein, I change that overall structure every single change. Why does this overall structure change as the primary structure changes? And the answer to that question is due to the fact of the chemistry of the R groups of the amino acids that you saw last time. Every R group has a unique, every amino acid has a unique R group, okay? And each R group has a unique chemistry. Some of them are similar. We see, for example, that aspartic acid and glutamic acid both have carboxyl groups. One has the carboxyl group on a little longer piece than the other one does, so we can imagine it might stick out a little further, et cetera, and it does, okay? So the chemistry of each one is unique. And it's the interactions that happen within a protein as a result of those different chemistries that give rise to different structures. Imagine, if you will, that I have a glutamic acid side chain over here, which is a COO- at physiological pH. And I have an aspartic acid over here that has a COO minus at physiological pH. The structure of this protein that contains these is going to be affected by the fact that both of these repel each other. Right? Similarly, if I have a positively charged amine from lysine over here, and I have a negatively charged carboxyl from glutamic acid over here, the structure will be affected by the fact that they attract each other. So we're going to see that there's several levels within a protein structure. There's several levels, attraction, repulsion, and so forth, that actually give the protein its overall shape. All right? All right, so that's very, very important. So the primary structure of a protein is important. How do we make proteins? We make proteins within ribosomes. Everybody knows that from basic biology, basic high school biology, actually. Ribosomes are where the proteins are made. Somebody asked me in an email, do we have, are we here? Yeah, come on up. This appears to be frozen as best I can tell. Okay, thanks. How are proteins made? They're made inside of a ribosome. I know, somebody sent me an email and said, are there proteins that are made outside the ribosome? And the answer is basically no, not inside of our cells. But there are some small peptides that, in fact, are made apart from the ribosome. We'll talk about one later called glutathione. Glutathione has 3 amino. Oh, looky there, you made me happy. It takes a little to make me happy. While you're doing that, I'm going to check my camera to make sure that we're lined up here while I keep talking to everybody else. So, excuse me. This row always gets it, so if you guys want to not have to adjust everything, just don't sit in that row. I'm not saying you're always welcome to sit in that row. Okay, if we are set, is the audio set with that also? Okay, great. Thank you so much. All right, magic. Applause, guys. We have video. Now you have to learn all this stuff, right? All right. Where was I? Uh-oh. Oh, I know. No, that should be working. Oh, he put it on this. That's what. Oh. Okay. Well, that's all right. Let me work out this. Let me change the thing then. There. And there. Okay, that's fine. All right, there we go. We got control. All right, so peptide bonds. Peptide bonds are the fundamental bond stabilizing primary structure of a protein. We make a peptide bond by putting together the alpha amine of one amino acid with the alpha carboxyl of another. Okay? That forms the peptide bond that you can see in the structure there. That involves loss of water. Water is lost in making a peptide bond. Bear with me? Okay. Proteins are made in moving from the amino end of a protein to the carboxyl end. Meaning that when you see the thing up there that says N-terminus on the lower left portion, then we'll give me the cursor. The lower left portion, you see N-terminus. That's the amine end, or the amino terminus of a protein. The carboxyl terminus on the other end, that's the C-terminus. And proteins are made by growing that C-terminus. The next amino acid will go onto the C-terminus, and then onto the C-terminus of that, and the C-terminus of that, et cetera, et cetera. So proteins grow from the amino to the carboxyl end. That's what we say. Okay. This is a polypeptide chain containing several amino acids. We can imagine, whoa, that's what, maybe about 15, 20 amino acids right there. There's a lot of different possible ones that we could have. But proteins are not made randomly. They're made according to a specific dictate. And that dictate is found in the coding region of the DNA for that protein. The coding in the DNA is translated into protein sequence using information in the genetic code. We'll talk about that later. Okay? So, primary structure of protein is simply a linear sequence of amino acids. These are joined by peptide bonds, as I have said, and it's translated, as I said, using the genetic code. Synthesis begins at the amino end and terminates at the carboxyl end. As I said earlier, and I want to pound this in your head, every property of a protein arises from its primary structure. Every property of a protein arises from its primary structure. Okay? This shows the primary structure of a short peptide. It has 6 different amino acids. And at first glance, when you look at this, you say, well, that's a lot of atoms that's up there. As you look at this, what strikes you? Anybody, somebody. Tell me what do you see when you look at this? What do you see in this structure? What do you notice? You notice a ring, okay. What else do you notice in here? Any common things that you see as you look at this? Yeah. Alternating R groups on each side. That's one of the more advanced ones. Yeah. Alternating R groups on each side of the chain. We'll see that important. Yes, Arisa. Every carbon is attached to a nitrogen. Okay? Every carbon is attached to a nitrogen. It goes from amine to carboxyl, so we see the amino end on the left, the carboxyl on the right. That's generally the convention we have when we write a protein sequence, that the amino terminus on the left, the carboxyl terminus on the right. Okay, these are all very good. You're right. The amino terminus is on the left, the carboxyl terminus is on the right. The amino terminus is the place where we have the only free alpha amino group. All the other alpha amino groups get gobbled up in making peptide bonds. So if I ask you on an exam, how many free alpha amino groups will you find in a protein, the answer is uno. Okay? You get one, only the very first one. You can probably figure out pretty quickly how many free carboxyl groups you have. Uno, right? Only the last one, the one at the very end, because all of the free alpha carboxyl groups get joined up in making peptide bonds. So are those the only amine and carboxyl groups we find in a protein? No, because we have R groups, right? Now, I've just told you a real cool secret about proteins. When you go to calculate the charge of a protein, which you're going to do, okay, when you go to calculate the charge of a protein, I've just simplified a lot of things for you to determine the charge. The only parts of a protein that can ionize are the alpha amino group, the alpha carboxyl group, and any ionizable R groups. All those other alpha amino groups and alpha carboxyl groups don't ionize. The ones in the middle don't ionize. They got gobbled up in peptide bonds. They no longer ionize. So I want you to keep that thought I just gave you planted in your head. So we've simplified understanding the charge of a protein. Okay. There are the peptide bonds in that protein. The peptide bonds occur, as you see, every third bond. And we can look at that middle part as being the sort of what I call the backbone of the protein. The backbone of the protein has bond, bond, peptide bond, 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 peptide bond, 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 peptide bond. And somebody noticed over here that the R groups are on opposite sides as they alternate. There are the alternating R groups. The first one on the lower left, the second one up above, down, up, down, up. Now that's not random. There's a very good reason that happens, and we'll see that right here. The peptide bond has an interesting property. It's a single bond. You saw it in the structure. Look at those peptide bonds. That's a single bond that's there. But in the next slide, I show it as being resonant with a double bonded structure, meaning that it has some character of a double bond. And I hope you remember from your organic chemistry that double bonds don't rotate. Single bonds can rotate freely, double bonds can't rotate. What does that mean about the peptide bond when it's a double bond? It means it's frozen. Can't rotate. Well, if you have a double bond, one of the things you learn in organic chemistry is what? You have cis and trans configurations because of the fact that you can't rotate. And in fact, that plays out as we see the overall structure. The alpha carbons are oriented around that double bond in a trans configuration. We'll get to the R groups in a second. The alpha carbons are oriented around that double bond in a trans configuration. There's a very good reason for that, as I said. The reason is here. We can see the same backbone structure, the peptide bond in red in the middle. We see it in the trans configuration on top, and we see it in the cis configuration on the bottom. You can see those blue balls there. Those blue balls are, in fact, the alpha carbons. Okay? On the top structure, they're in the trans configuration. In the bottom structure, they're in the cis configuration. Which one is going to have more problems with steric hindrance? Well, the one on the bottom. Okay? When we look at proteins, we see a very strong preference for trans orientation of those. Well, since the alpha carbons are in the trans configuration, and since the R groups are attached to the alpha carbons, the R groups, by extension, are in the trans configuration. Does that make sense? Question back here. Oh, okay. I read your mind. So the trans configuration, so trans always relates to molecules around a peptide bond. The bond itself doesn't have cis or trans, but it's the arrangement of materials around it. So when I talk about alpha carbons being in a trans configuration, they're oriented opposite instead of being on the same side of the double bond. So the alpha carbons are on the opposite sides, and because the alpha carbons are what contain the R groups are on the opposite side. So every other one rotates. Okay? Atoms hate steric hindrance. They avoid it. And that's why we don't see that happening much here. It happens occasionally, but we don't see it much. Well, this now shows a polypeptide in a little bit more up close and personal detail. It's the same one that you, no, actually it's not the same one you saw earlier, but it's similar to the one you saw earlier. All right? And you can see now each peptide bond has been labeled with a little blue box around it. That little blue box around it defines what we call a plane. Because a trans double bond creates a flat planar structure. The cis and trans actually does that. So it's flat. And because it can't rotate, it stays flat. Now, if we look at this, what we see is that we have a flat structure on the left. Okay, the farthest left rectangle that's there. Attached to it, we see a single bond, and we see a little arrow rotating around, meaning that that's a single bond that doesn't have any double bond character. It will rotate. We see adjacent to it another single bond that can rotate. And then we hit another peptide bond with the blue rectangle. Now, that shows you, I'm going to show you with my nose and my hands what that's trying to tell you. Imagine, if you will, this is one of those peptide bonds, and this is the other of those peptide bonds. My thumbs are the single bonds, and my nose is the alpha carbon. Okay? What this means is that my hands, which are the planes, remain flat, but that rotation around those single bonds can occur. Everybody got that? Okay. So when we think about the backbone of a protein, there are three relevant bonds. The peptide bond, and each of the single bonds to the alpha carbon. We can also think about the backbone of a protein as containing the two atoms of the peptide bond separated by an alpha carbon. Peptide bond, alpha carbon, peptide bond, alpha carbon, peptide bond, alpha carbon. That is the backbone of a protein. Now, this observation that I've just given you, okay, is essential for understanding fundamental protein structure. That flat structure places a lot of restrictions on possible structures a protein can have, as we will see. Questions about that? Yeah, I was either so clear or I was so confusing that everybody's going, okay, I'm hoping that means I'm clear. There's the rotation, and now this is this guy up close and personal. In this case, the central thing here is the alpha carbon. It has the equivalent of the blue rectangles on either side of it. In the middle of it, we see those two regions that can rotate. Those rotatable bonds have names. They have angles that we call them. They're called rotational angles. Those rotational angles are known as phi and psi. There's a phi angle, and there's a psi angle. And I've got good news for you. You're not going to need to memorize which one's which. Yeah, right? You should know what they are. You should know where you find them. But I'm not going to draw you a structure and say, okay, guys, is this a phi or is this a psi? Because you wouldn't really be telling me anything important as far as I'm concerned. We're not doing biophysics here. We're understanding principles of structure. Everybody with me? Okay. All right. Now, you see the peptide bond has something associated with it called omega, and that's a third angle that's there. We're not going to talk about omega. Omega has some significance, but we're not going to talk about it. Remember, it's not really a rotatable angle, okay, because it's got that double bond character. Well, there was an Indian scientist named Ramachandran. And Ramachandran was way ahead of his time. Many people felt he should have won the Nobel Prize. He was way ahead of his time because he realized that if you took these planes and these planes and you started rotating them, it's not quite like my hands. There's not nearly as much room out there in three-dimensional space as my hands would indicate. Steric hindrance is a problem. Steric hindrance is a problem even out here in rotation. Steric hindrance is always a consideration. We try to bring atoms close together. Ramachandran realized that there would be problems, there would be angles that would not be stable corresponding to rotation of certain phi and psi angles. So he asked a computer program, knowing the dimensions that you know of the peptide bond and all the various atoms that are in there, and we know all those dimensions, and you start rotating them through space, and you make some certain assumptions about the energy associated with interactions, predict the regions that will be stable and predict the regions that will be unstable. He created what was called a Ramachandran plot. That's what you see on the screen. Ramachandran plot plots the stability through 360 degrees, I can't do that with my hand, but 360 degrees of rotation of this one, and 360 degrees of rotation of this one. That's why it goes from plus 180 to minus 180 on the top, and minus 180 to plus 180 on the bottom. And when he did that plot, the computer said, well, the darkest areas here will be the most stable regions. These are the angles that will give the most stable structures for a protein. Okay? Now I'm going to come back to this plot in a couple of minutes and describe more about it. But I want you to see what that is and how he made that assumption and calculation. He turned out to be very, very correct. Very correct. Okay. Let's talk about structure. We've been talking about structure. We're moving on from primary structure, which is the sequence of amino acids, to talking about secondary structure. Secondary structure, I'm going to define it for you here, arises from interactions between amino acids that are close in primary sequence. Secondary structure arises as a result of interactions between amino acids that are close in primary sequence. An amino acid interacting, let's say an amino acid glycine, that interacts with an amino acid tryptophan 3 or 4 amino acids away, would be participating in a secondary structure. If I have an amino acid glycine here and I have a tryptophan, let's say that's 300 amino acids away, when they interact, that's not going to be secondary structure because they're too far away in primary sequence. Does that make sense? Okay. Interactions of amino acids that are close together give rise to regular structures in many cases. One of those regular structures you see on the screen. It's known as an alpha helix. The alpha helix was discovered by Linus Pauling. Linus Pauling won the first of his 2 Nobel Prizes for the alpha helix. Now, what's the red thing on the side? That's an alpha helix in front of Linus Pauling's boyhood home. Okay? That's a cool structure. The alpha helix is one of the most fundamental structures of proteins. Okay? Linus Pauling, I'm sure all of you know, was the most famous scientist ever to graduate from Oregon State University, graduated in the 1920s with a degree in chemistry. He's the only person in the world ever to win 2 unshared Nobel Prizes. The first of them was this one. Okay? The man was easily one of the most intelligent, genius people of the 20th century. Many put him in the top 3 of all scientists of the 20th century and probably in the top 10 of all scientists who ever lived. Amazing man, right here. Same place where you're studying. The alpha helix has within it these dotted lines that you see, and those relate to the interactions that I'm talking about. Hydrogen bonds stabilize alpha helices. Hydrogen bonds stabilize alpha helices. Now, a lot of classes, people go through exactly how many amino acids and exactly the dimensions and exactly how many turns per twist and so forth. And again, that's nice trivia information, but it doesn't tell us anything nearly as much as that structure tells you. You know from what I told you that it's arising from interactions that are close, and by close, I'm talking about less than 10 amino acids away, usually about 3 or 4. And if I break those hydrogen bonds, then I would, in fact, destabilize the alpha helix, and I would destabilize the structure of a protein that contains it. Another important structure discovered by Linus Pauling was that of the beta strands, or as some people call them, beta sheets, and they're actually different things. A beta strand is one alternating sequence. So we see here parallel. If you look at the structure on the left, that's a beta strand. Beta strands are commonly paired with another beta strand, and the structure on the right paired with it makes a beta sheet. So it's like if you have thread, you have one thread, and you weave them together, you have a sheet, right? That's literally what's happening here. So a strand is one thing, a sheet is made up of many strands, okay? Well, those strands interact, as you can see, and you can see those same dotted lines, and you can see hydrogen bonds are stabilizing the beta strand. What is a beta strand? A beta strand is interesting. A beta strand is an alpha helix, except for it's flat. An alpha helix has a coil. It has a 3-dimensionality to it. A beta strand only has 2 dimensions. Doink, 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 doink. It doesn't have that full rotation. Okay? People talk about pleated sheets or pleats in the strands because they form bends very much like pleats of a curtain or something like that does. Okay? Well, there's 2 different ways of arranging them in space. One is in the parallel orientation. Amino to carboxyl going up and amino carboxyl going up interacting, that's parallel. Amino carboxyl going up and amino carboxyl going down, that's antiparallel. So it depends on how they're oriented as to whether they're parallel or antiparallel. Hydrogen bonds. You see the arrangement of hydrogen bonds a little bit different in the antiparallel than in the parallel, but for our purposes, the hydrogen bonds are stabilizing that. There's an example of an antiparallel with a real live ribbon diagram that people use to show protein structure. There's a more elaborate one. Some of these get really beautiful. Okay? Now we put atoms in and we start seeing those blue rectangle boxes for the labeling where the peptide bond planes are. Yes? Question? Will antiparallel be more stable than parallel? No, not necessarily. Not necessarily. So it's actually that's a simplification that's there, so that it's actually not you shouldn't infer too much from that structure. Okay. Okay. So in this case, if I gave you this on an exam, would you tell me you're looking at parallel or antiparallel? I hear both. It's parallel. Okay. Those nice straight bonds that we saw look like you only had those really straight things when you were doing parallel, and the uneven ones when they were unparalleled. Well, the reality is in the real world there's a little unevenness that's there. The clue that gave it away was the fact that the amino ends were at the same end and the carboxyl ends were at the same end. So this is a parallel structure. I said some of these are beautiful. Okay? One of my students drew this. And it depicts real life structures we find in proteins. We see that those sheets can have many threads, many strands. And we also see that alpha helices can arrange themselves in some ways that gives some really beautiful structures as well. We'll see many examples of these as we go through. You also see the word up there called supersecondary. What the hell is supersecondary? Well, I use that term. I'm kind of alone in this. A lot of people don't use that word. Okay? I like to use it because how did I define secondary structure? I said they were interactions between amino acids that were close to each other in primary structure. If I do an antiparallel, this guy that's down here at the bottom is not close to something that's 10 amino acids away. It's a little further away. Still pretty close in primary sequence. So I call those supersecondary. So these things that you see on the screen, when you see a sheet, I refer to sheets as supersecondary structures. Okay? If I had just a strand, I would be talking only about secondary structure. Okay. Well, let's go back to the Ramachandran plot that we did before. The Ramachandran plot now makes interesting sense. The regions that we see of stability on the Ramachandran plot that were theoretically predicted correspond precisely to the angles associated with beta strands on the top, right-handed alpha helices on the bottom, and left-handed alpha helices on the right top. Left-handed alpha helices are not common at all. Now you might say, what's left-handed, what's right-handed about a helix? If you really want to answer that question, come in my office and I'll show you a cool example. But suffice it to say that two strands, or a strand, can wind about itself in two different ways. Imagine, if you will, that you look at a coil and the coil is going away from you. There's two different ways it can coil away from you. It can coil away from you in a clockwise direction, and it can coil away from you in a counterclockwise direction. And no, it's not independent. Either end will have the same thing. Okay? If it coils away from you in a clockwise direction, that's a right-handed helix. If it coils away from you in a left-handed orientation, if it coils away from you clockwise, it's right-handed. If it coils away from you counterclockwise, it's left-handed. Clockwise, right-handed, counterclockwise, left-handed. Okay. Well, I'm sorry, I can't hear you. It's a good question. So Ramachandran plots are useful for theoretical projections, but as you get into biophysics, as some of the people in here are, one of the things that you discover is that when we see a protein structure, these beautiful 3D structures that people show, biophysicists determine those. And how do they determine them? Well, they have to model. They have to take data and see if models that they build are consistent with the data that they have. And the Ramachandran plot provides limitations on the models that they build. So it actually helps them to build the structure of a protein that they're analyzing. So they do have actual real-world applications as well. Secondary structure is important. Some proteins have primary structure and secondary structure, and that's about the only kind of structure that they have. We call those proteins fibrous proteins because they form fibers. Okay? The most abundant protein in your body is a fibrous protein. It's collagen. The most abundant protein in your body is collagen. How many knew that before I said that? A few? Not very many. Okay. Collagen's the glue that holds you together. There are 29 different forms of collagen, of human collagen. 29 different forms of human collagen. Okay? Forms cartilage, helps stabilize bones, connective tissue of your skin. Collagen's found everywhere. And collagen is a fiber. You can see some of those fibers on the screen in the picture on the right. It's found in connective tissue, as I said. Another common fibrous protein we find in our body is keratin. Okay? Keratin is found in nails. It's found in hair. Okay? And it doesn't really have other types of structure in it, as we'll talk about in a bit. Another common protein that has pretty much secondary structure is that of fibrin, which is the protein that's important for making silk. Okay? Now I want to start talking about these in terms of collagen. because collagen illustrates a couple of interesting things to us. Okay? What you see on the screen is the partial amino acid sequence of collagen, of one of the collagens. There's 29 different ones, but this is one of the collagens. And looking at it, I could ask you some questions in terms of, well, what do you notice about it? And you're probably going to notice what I'm going to tell you. I'll ask you since I said I would do it anyway. What do you notice? The what? They all bonded together what? Every third was a glycine. Very good. Every third amino acid there is a glycine. You guys get the harder ones first. What else do you notice on there? Can't hear you. Ends in two aspartic acids. That's an interesting observation, but that's not the end of the protein, so that's just random there. Yeah. Anything else that's abundant in that protein? Anybody see proline in there? Anybody see HPR in there? Which amino acid is HPR? Hydroxyproline, but hydroxyproline is not one of the 20 amino acids found in proteins, but here's this guy, and it's in protein. How'd it get here? Last lecture, guys, how'd it get here? How did we get hydroxyproline in there? What's that? No, guys. Anybody remember the term post-translational modification? Oh, yeah. That was the end of the lecture, though, so everybody's brain turned off by that point. Post-translational modification takes an amino acid and covalently modifies it after it's in the protein. That was originally a proline, but some enzyme came along and put a hydroxyl group on it and made it hydroxyproline. If we look at that, we see a lot of hydroxyprolines. Okay? A lot of hydroxyprolines. We also see a fair number of prolines. Now, that may be surprising to you. Why is that surprising? Why would that be surprising based on what I've said? What does proline do in a protein? It's not Friday, folks. What does proline do in a protein? What property happens when a proline is found? Yeah. It creates a bend, yeah. And here's this beautiful coil that we're talking about, and now it's full of all these things that cause bends. How in the hell do you have a coil that's full of an amino acid that makes bends? Ahern's shitting us again, right? No. Okay. The answer is rooted in the fact that every third amino acid is a glycine. What property did glycine have? Relative to its R group. What's that? It's a hydrogen. Glycine has the smallest R group. R groups are involved in steric hindrance, right? Which one's going to be able to accommodate more room of other things than any other amino acid? Glycine. Glycine is allowing that helix to happen. So proline is not, in this case, making bends every time it appears. It's actually able to be part of a helix thanks to glycine. And that's why glycine's in there every third amino acid. Okay. There's those prolines. Okay. By the way, I should say something about those hydroxyprolines. Oh, damn it. Those hydroxyprolines, okay, are there for a reason, all right? It turns out that the addition of those prolines helps to stabilize the protein thermally. What does that mean? Thermal stability. What's thermal instability? What would that mean? Heating it up would destroy it. So thermal stability would mean the opposite. Hydroxyproline allows the protein to be stabler at a higher temperature. The higher temperature in this case is your body temperature. If you didn't have those hydroxyprolines in there, your collagen would not be stable at body temperature. That's not very hot, right? The hydroxylation of proline requires a vitamin. It requires vitamin C. If you are deficient in vitamin C, you will be unable to hydroxylate your proline. If you're unable to hydroxylate your proline, what's going to happen to the glue that holds you together? It's not going to be thermally stable. Right? That condition is known as scurvy. You get scurvy if you are deficient in vitamin C. Today, in today's world, virtually nobody is deficient in vitamin C in the industrial world. Okay? That's not the case in other places. Okay? The way they discovered this was with pirates. Pirates used to go out on the ocean, and they would go out these big, ugly-looking, smelling things that you can imagine, hulking things, and they would go out to sea for a full year, and all they would eat would be salted meat, and they got essentially no vitamin C, and when they came home, they were either dead, that's where burial at sea came from, or falling apart. And it was not really fully realized until about the 1500s that citrus fruit could keep that from happening. And even then, after it was discovered, there were still a couple million sailors who died on the oceans of scurvy. Okay? Yeah, not a good way to go. Okay. There's that. Oh, one last thing. Lysine. There's one lysine on there. Nobody noticed I have one lysine. Lysine occurs in collagen infrequently. But it's important because lysine on a strand over here can interact with a lysine on a strand over here. It turns out that collagen gives stability because the strands are coiled together. They form a coil of coils. So we have a coil, and that coil is coiled with another coil, just like you braid your hair. Well, if you braid your hair and you don't put a rubber band on the end of it, what happens to your hair? It falls apart, right? If you put collagens together and you don't link the lysines together, the strands can't stay together. It will fall apart. So lysine is also needed for the structural integrity of collagen because it's literally the rubber band that you put on your hair. In this case, it's a covalent bond that forms between lysines on different strands. Keratins, great example of the horns here. Okay? They're fibrous proteins. There are about 50 of them in humans. They form subcellular structures in the cytoskeleton as well as extracellular structures, as you can see here. They form hair, nails, and horns. Oop, I'm going fast. Didn't mean to do that. Fibroin. Fibroin is the component, the protein that's found in silk. And fibroin I show you as an example because it's primarily beta sheets. Others form helices of sorts. And if we look at the sequence of fibroin, it's a repeating sequence like you see on the screen here. This repeating sequence has glycine, not as every third amino acid, but as every other amino acid. This is a very, very glycine-rich protein. Okay. So, let's talk about, we already talked about alpha helix. We've talked about the beta strands and the beta sheet, right? Something else to consider, turns. Not all proteins are fibrous. Sometimes that helix gets started, and then we have a proline that's not next to a glycine that makes that bend that we talk about. And we have a bend, we have something like what we see on the screen here. They're called reverse turns because they're reversing the direction that the chain is going in. Okay? These turns vary in the number of amino acids that comprise them. You see one here that has 4 different amino acids, N, N plus 1, N plus 2, N plus 3. The number of amino acids in the middle that separate them distinguish them. So there are 5 different ones depending on how many amino acids we put in the middle. We don't need to worry about the specific structures. There's 5 different kinds of reverse turns, varying in this, basically the angle or the size of that turn. The last thing I'll talk about is another type of helix that we find inside of cells called a 310. Okay? All right? 310. It gets its name for the fact that it makes 3 turns in 10 amino acids. You can see it's a beautiful structure that you can see on the screen there. the carboxyl groups, actually, no, I won't say that. I'll just leave that be. So a 310 helix. Okay, questions? Somebody told me today, Ahern, you go fast. I do. Try to slow down. If you guys don't slow me down, I just keep going. Pull my string. All right. Well, the last things I want to talk about today relate to properties of those amino acids. We've talked about them, but I now want to show you a little bit of them. One of them is known as hydropathy, or you can think about it, the non-polar nature of the amino acid side chains. Hydropathy will come into play in a minute because it helps us to determine where certain amino acids are found in proteins. The plot you see on the screen, however, is not hydropathy. The plot you see on the screen is a measure of the tendency of any amino acid to be in any of those structures, an alpha helix, a beta strand, or a reverse turn. The larger the number, the greater the tendency. Now, you'll notice that no amino acid has a 0 for any of those, meaning we find every amino acid in every one of those structures. Okay? Some are more likely to be found in one than the other. Okay? Proline, for example, we don't see found very much in alpha helices. We don't find it found very much in beta strands, but we find it a lot in turns, and that's what I've already told you, right? The inflexible nature of the proline makes those turns. If we look at a couple of these, for example, we see alanine with a 1.41 and glutamic acid with a 1.59 like to be in alpha helices a lot. Well, how do we know what if I have a sequence of amino acids, how do I know what that sequence of amino acids, what structure any amino acid is in? Well, I can't tell absolutely, but I can frequently predict secondary structure from sequence. A computer program can do this very easily. Because if I have 3 amino acids in a row that have a high value for a particular structure, they're likely found in that structure. So, for example, if I had a sequence of amino acids in a protein that was alanine, glutamic acid, followed by, I don't know, pick one here for me. Let's say arginine. They're all values over 1, okay? They're all consecutive. They're probably part of an alpha helix. Okay? So the computer can go through and it can map out for you very quickly what regions are in alpha helix and what regions are not in alpha helix. There's some that's propensity for beta strands. There's some with a propensity for turns. Glycine, you see, likes to be in turns. It allows for a lot of things to happen in turns. Well, the hydropathy, which I jumped the gun on the last slide, is actually a measure of the nonpolar nature of an amino acid side chain. The higher the hydropathy value, the more nonpolar the side chain. Not surprisingly, the ionizable amino acids are at the bottom because they've got charge. The other ones are up at the top. We see some in the middle with about, glycine, for example, is close to 0. We can use those values, tell a computer program, look across this protein and calculate the average for every 3 amino acids in this protein, calculate the average hydropathy value. And when you do that, you see for a given protein, you see this. Hydropathy is a measure of the nonpolar nature of the R group of an amino acid. The higher the value of hydropathy, the more nonpolar it is. Okay? Am I doing that in time? I'm running out of time. Okay. Finish in just a second. Okay? Everybody got this? Now I'm going to come back. I'm going to talk about this next time. But before that, you know what we have? We got to get it through here. It's called a little protein molecule. Plastic. All right, I'll tell you what. We'll start with a song next time instead of finishing with one today, okay? Begin with a song next time.